will discuss lens-free microscopy. This is a new mean of microscopy that we are developing here in Grenoble since 5 years. And this is one of our latest prototype of lens-free microscopy. I guess most of you very familiar with standard microscopy or with a confocal microscopy. This is a new mean of microscopy and it's very minimalist. That is, what remains from a standard microscope is the illumination. We use an LED and the CMOS sensor. And as you see, there is no lens. That's the name of the microscope. It's what defines the microscope. There are no magnification, no lens anymore. And basically what we do, when we take a smartphone, here is the small camera that you use to take pictures, and we remove the lens. And what remains is the small CMOS sensor. And we can take the LED from the smartphone, and you can build a microscope with 3D printing. So basically, what we do, we take a smartphone and we remove the phone. It's no use. And then we put the object, what we want to look at. These are the cells, these are the bacteria, these are the viruses, these are 3D cell culture on top of the CMOS sensor. And now the question is, what can we see with this device? And now I will show you what we are seeing with this device. And as I told, we are working since, well, it's still, can still be used. And as I told, we are working since 5 years now on this device. And I will make a chronological story of our development. And I will show you that we are able to detect bacteria, 1 micron bacteria, that we are able to detect single viruses. This is H1N1 viruses, adenoviruses, only 70 nanometers. This was the beginning. We start in 2010 with bacteria, viruses. And since a few years now, we are very much interested into cell culture in 2D, in 3D. And there is a PhD works right now to look at tissue slide. And I will explain you how we can detect viruses, bacteria with this very simple device, and how we can make pictures of cells and tissue slides. Okay? So to detect bacteria, what we do, we put a droplet full of bacteria on top of the slide. Here's the CMOS sensor, and here's the light. And what we do see when the bacteria is in the droplet, we see nothing. It doesn't work. Because a bacteria is only 1 micron of water into a droplet of water, you will see nothing. But what we have discovered is that when the bacteria, it goes into the meniscus of the droplet, there is a very strong signal for a single bacteria. Signal to noise ratio is 100. But it's no use because you have to bring the bacteria in the meniscus, and even with the capillarity, it's very difficult. So the way we found is to make a very thin weighting film of water on top of the bacteria. And this will make a lens on top of the bacteria. And this will give you the light at the sensor. So here are the bacteria on the slide. The sensor is 2 millimeters away from the bacteria. Here's the 1 micron bacteria. But then there is a very small lens on top of the bacteria, and it will shine the light 2 millimeters away down to the CMOS sensor. And this way we are able to detect a single bacteria. And this is the way we proceed. The trick is to put soap into the sample. We put 1% of soap, and when the sample evaporates, what remains is the soap. And here you see the soap on top of the bacteria, and on the top of every single bacteria, there is this microlens. It's a self-assembled microlens, and we are able to detect single bacteria with your smartphone, basically. And we have been working on the modelization of this lens. We have a physical model of this lens, and what we understood is that the thinner the weighting film, the thinner the soap film, the smaller object we will be able to detect. If you want to detect a bacteria of 1 micron, you have to make a film of 1 micron thickness. But then we understood that if you want to look at viruses, then you have to make a film of 100 nanometer in thickness. And this time we are not using soap anymore. We are using a polymer. It's called PEG 600. This is the stuff people they smoke in the e-cigarette, you know. We use it to detect the viruses. Because with this PEG 600, we're able to make very thin weighting film of 100 nanometer, and now you can detect, this is a mixed sample of bacteria. There were 500 nanometer bits, and granulovirus 300 nanometer. These are viruses that you can buy here at Botanic. This is why we look at these viruses. And in collaboration with a lab in UCLA in Los Angeles, the lab from Professor Heidegan-Oskan, we have been able to detect single viruses of H1N1. They are pretty large. Well, they are 200 nanometers. And the smallest one that we have been able to detect is a single adenoviruses of 100 nanometers. And this was a kind of tour de force, and this has been published in Nature Photonics in 2013. So with this device, we paved the way maybe for a new means of diagnostics to measure the viral load assay or the bacterial load assay. And then in 2012, we have been looking at cells. And now cells they are much larger, and we want to make picture of the cells. Before, we only detect the object, but we cannot make a picture of the viruses. We cannot make a picture of the bacteria. But the cells, we want to make a picture. And with the cells, we are not using anymore the soap. We are not using anymore the polymer from the e-cigarette. We are using physics. I will try to explain you by hand how it works, because there is no magnification. This is the LED. These are the cells. And 2 millimeter away, there is the sensor. And what we are looking at is these are diffraction patterns. So what are diffraction patterns? If you throw a stone into the water, you will see rings, wavelets of water around the stone. And if you shine light on the cells, the same happens. You see rings of light around the cells. And these rings, they are much larger than the cells. They are 200 micron in diameter, 500 micron in diameter, and you can make the picture of these rings without lenses. And this is what we do. This is the picture of the cells. I mean, in the diffraction mode. This looks like nothing to you. And then we have computer to make the shape of the cells. And what we do is, when you look at the stone into the water, if you make a movie and you play it backwards, you will see the stone appearing. We do the same with the mathematics, with the physics. I will show you the picture. It's more clear. So this is the picture that we get. These are stem cells. You cannot recognize them. But you see the rings of light around a single cell. And then, thanks to computer, we can make the picture of the cells. There were cells here, and after computation, you see the picture of the cells. And since the device is very small, you can put it into the incubator. This is the commercialized microscope, lens-free microscope, since last year, a year ago. So this is the Petri dish. This is a 35 millimeter Petri dish. The cells, they're here. The sensor is here, and the LED is here. And the whole device is that small. So you can put it in the incubator, and now you can make a picture every 5 minutes. And then you can look at the cells. You can see a stem cells dividing here. This is a 28 hours movie, so it's pretty much accelerated. Yeah? These are neurons. So you can see a connection. The resolution is about 1 or 2 or 3 micron, but it's enough to see the dendrite, and it's enough to see that these two neurons are getting connected. Here's dendrite that is useless, it is removed. You can see another dendrite there. It tries to connect with these cells. So it takes like 20 hours to try to connect, and it will not connect, so it will get back. And so other example, these are cells from the retina. But what's very important with this device is that the field of view is much larger compared to a microscope. That's a regular microscope, it will look at this dimension about. Here, we look 6 millimeter by 5 millimeter. And in total, there are 10,000 up to 20,000 cells at glance. Of course, we can do a lot of computation. We can measure the surface occupied by the cells. We can count the cells. We can draw kinetics. We can measure the number of cells, the occupied surface. We can measure the speed, the adhesion of the cells, the length, the area, the number of neighbor cells, etc. And we can plot scatter plot of this matrix along times. So this is a 2 days experiment. And this plot, there is one measurement every 5 minutes. There are 10,000 cells, so we end up with a data of 1 million of measurements for every single cell culture. This is a drug assay, for instance. This is the cloud of 3,000 cells. These are the living cells. Then we introduce the drug at this point. And we see the kinetics. All the cells, they will experience the drugs, they will die. But what's very interesting, since we have a lot of statistics, we see the 200 resistant cells. We have 3,000 cells, they do experience the drugs, but remaining are 200 resistant cells. And because we have this large number of cells, we can point this low resistance. So these are measurements at the cell population, but we want to look at heterogeneities, subset population, and we also want to look at single cell measurements. And to do single cell measurement, first we have to do cell tracking. We have to follow every single cells, and with a cell tracking algorithm, we put colors on every single cells. And again, we have a very large field of view. So these are UV cells for 3 days, and you see how it evolves. And then we can track every single cells. And for instance, you can see that the cells, they can dance with 2 and 3. And then with 4 and 5, it's a mess. They cannot dance. And this to show that we can measure every single track. So we have a lot of statistics on the tracks of the cells. We can compile hundreds of cell tracks. And the measurement is as long as 24 hours, for instance. And then we can plot all the tracks of all the cells for 1 day. And then you see that the cell lines are very different one another. These are UV cells. They remain close one another. They never go from one point to another point. They like to remain together. These are fibroblasts. They don't like their neighbor. They go everywhere. And macrophages, they like themselves very much. They like to stay at the same point. And there is a subset of populations they want to see abroad. You see, they go far away. So these are the heterogeneity. You see different subset of population. And we can also measure all this metric at the single cell level. Here you see a single cell. I mean the mother. You will see the next division. It will divide, yeah, sure. And then you can follow every single cell among this very large population of cells. I've shown you various bacterial cells, and I will end with a tissue and 3D cell culture. So today, in the thesis of Sophie Morel, we're interested to use this technology to look at tissue slide. And if you remember the picture I used in the beginning, you throw one stone, you look at the ring, and then with the computer, you make the image of the cell. A tissue slide, these are millions of cells. So it's like you throw millions of stones into the water and try to make the picture of it. So it's a very difficult task. And we are very pleased that in the PhD work, we managed to do that. And then we get the color in addition. So this is a tissue slide of the intestine. And we take a snapshot with a single image, 4 millimeter by 3 millimeter. And then, of course, we have to make a mosaic of many pictures. We can zoom in. So you see the lumen, you see the gland there. And then you can zoom in again, and you can see every single nucleus. So this is to show you the resolution that we achieve with your smartphone. Okay. And this is another example. And these are the comparison with the microscope. And I want to finish with 3D cell culture. I mean, it's not truly 3D cell culture, but it's different in the sense that typically we do 2D cell culture, what I call 2D cell culture. These are the cells on a petri dish, on a substrate, on plastic. And I want to show you the difference of this cell line in particular when we look at with our microscope in 2D and on matrigel. So these are UV cells on 2D, okay? This is how they behave. What's interesting in this picture is to look at is because you have a very large field of view, at some point you see a kind of fluidic flow of the cells. You see they rotate around vortex. This is something that you will not be able to look at with a standard microscope. So this is in 2D. And now we put the same cell on a matrigel, and you will see the difference. Here we go. These are not exactly the same cells, but this is the same cell line. And you see how it evolves and how to assemble. They all, even make a square. It's very rare in biology. So we are not squared. And I will end my talk with a puzzling picture. Left, these are the UV cells that we have observed. These were one-day experiments. And the right, this is the cosmos. Here, every single dot is a galaxy. It is not a star, and it's a 3 billion simulation, 3 billion years simulation. And the kinetics and the shape and everything looks pretty much the same, yeah. And this is the end of my talk. Thank you.